good morning to the American viewers. I'm not sure how many Europeans and Asians are joining this meeting, so afternoon and probably evening for you folks. It's a pleasure to chair this session. I am Sabia Gan. I'm actually chair of the Chemical Applications Interest Group, and it is my task to organize a meeting like this, where I have the liberty to choose the topic. After the survey last year, we decided to choose the topic of antimicrobial substrates used in textiles. I think it's quite relevant, and I approached the speaker. So you already know the speakers. I'll briefly introduce them. But before I go into details of this topic, I think we should do the housekeeping rules. So this meeting is recorded, and if you don't want to be recorded, you should switch off your webcam as well as mute yourself. We also, in case you want to put up questions, it would make sense to send it on the chat itself and raise your hand. So we will go through all the questions at the end of each presentation, and then we go through all the questions. So we will have some time for the questions. Raise your hand, put your questions in the chat. What else do we have? I think that's pretty much it. I hope that we have a very productive meeting where we are all engaged. So let's move to the speakers. So today's topic actually is very interesting. When I decided to invite my speakers, I was not sure what they're going to speak, and I would say it was a coincidence that both my speakers, Dr. Ren and Mr. Santos, they wanted to speak on the topic of probiotics. I believe some of you, or most of you, have heard the term probiotics. It's the use of living, I would say, microorganisms, which is beneficial to our body, and especially probiotics are quite popular for dietary purpose. So you must have heard of this term probiotics. If your gut is upset and you want to recover it, you tend to use probiotics to help it, I would say. But I think the speakers are going to talk about it in detail. So in today's two presentation, we will focus on how we can use probiotics for textiles. And we start with the first presentation from Dr. Ren. She is a colleague of mine, and we have also collaborated together. She's a microbiologist, and she has a lot of experience in developing antimicrobial resistant materials, and I think she has a lot of experience in. Currently, she is focused on developing, I would say, bacteria, on how to use them for antimicrobial, I would say, substrates, and she has a lot of experience in doing national and international projects, and I'm happy to listen to the first presentation. So Dr. Ren, it's your floor, so feel free to share the slides and present. You have 25 minutes to speak, and around we will follow up with 10 minutes of questions later on.
Yeah. Thank you, Sabia. Can you see my screen? Yes, can. Huh? So, yeah. Hello, everyone, and my name is Chunren. And first of all, I would like to thank Sabia and all the other organizers for inviting me to present our work here. And as Sabia already introduced, I have a background in microbiology, and I did my PhD at ETH Zurich in biotechnology. And currently, I'm a group leader at the Swiss Federal Laboratories for Material Science and Technology, in abbreviation Empa. And I don't know how many people know about Empa. I just want to give a few words about our institution. Right? I cannot move. Yes, I can. So Empa is a government-owned institution. It belongs to ETH domain. So at Empa, we mainly work on material-related research and development. And my team, my group, is mainly working on studying interactions of bacteria and materials, and try to develop antimicrobial, antifouling materials, surfaces, and solutions, with the goal is to fight against antimicrobial resistant problem. So my team is really very interdisciplinary, including scientists with a background in biology, chemistry, or material science for sure, and also some physics even. So as I already mentioned, antimicrobial resistance, in abbreviation AMR. So why we are interested in this topic, why Empa spend so much resource on this AMR topic. So AMR by definition refers to microorganisms, including bacteria, viruses, fungi, and parasites, and to resist the effect of antimicrobial drugs. So okay. Problem 2 is, so today I will mainly focus on bacteria. So bacteria are everywhere on Earth. So they are super important for our ecosystem and also for our health. So in nature, most of bacteria exist in the form of biofilm. Means bacteria, they don't fly flowing around, but they like to adhere to a surface and then form a bacteria community, and namely biofilm. So we also know on our body, there are more than 3 times more microbial cells than our human cells. So normally these microbial cells and human cells, they can coexist in harmony. So this is, they can happily live together. But once we have external, let's say, injury, or our immune system get compromised, and then the really have a problem with to go next. Yeah, then the bacteria, pathogenic bacteria, can invade to our body. All the commensal bacteria means bacteria normally exist on our body, for example. They are not harmful. They are there either with function or without function. But when our immune system gets compromised, this commensal bacteria can turn into a pathogenic bacteria. So in this case, then we need to treat this pathogenic bacteria with antibiotics, for example. Once we apply antibiotics, the AMR problem cannot be avoided. Why this? So normally, as I mentioned, can you see my cursor? Or the mouse? So normally the bacteria, they don't exist in a single cell level. Normally they exist in a community. And then some cells in this community, they can mutate and then generate resistance. And imagine when we apply antibiotics to treat this community of bacteria, the sensitive ones will be killed, and then the resistant one will survive. And then this resistant bacteria will further proliferate and then spread. So you can imagine when you apply antibiotics, normally they exist, like more or less balanced. But when you use antibiotic, the sensitive ones will be killed. Then you have mainly the resistant ones left over. Indeed, this problem has been already identified, decades ago. So even though antibiotics have saved millions of lives since its discovery, but we also soon realized they have a resistant problem. For example, like this methicillin, which was introduced to the market in 1960, And then 2 years later, the resistant Staphylococcus aureus identified. And even more like recent antibiotics launched into market, like this levofloxacin was launched in 1996. So in the same year, immediately resistant bacteria identified. So this resistant bacteria, they can spread in our surroundings through different pathways, like from human to animal, animal to environment, all this, interplay and interaction between all these stakeholders. This really makes the AMR really a huge topic in the moment. So in the moment, we consider AMR crisis is one of our global challenges. So this has impact on our society, patients, healthcare, and hospitals. And it's also predicted by 2050, we will have 10 million deaths caused by AMR, which is even higher than deaths caused by cancer. That's why we call AMR problem is a silent pandemic. So what do we do at Empa? So we try to develop innovative materials and solutions to fight AMR problem, mainly in 2 areas. So on one side, we want to develop biosensors, which allow us to rapidly detect pathogens or infections to improve the usage of drugs. And on the other side, we also want to develop novel materials to replace the classical antibiotics. So this includes nanomaterials, bioresponsive materials, and including also living materials. And today I will mainly focus on using living materials, namely probiotics, for treating for fighting against the AMR. So probiotics, as Sabia also mentioned at the beginning, I think many of you or almost all of you know. So these probiotics are defined as bacteria which can confer beneficial benefits to human beings or to animals, to whatever, when supplied with sufficient amount. So you find probiotics in our daily consumptions, like including like some yogurt or kimchi and many other food supplements. And this has been extensively studied and even applied in our daily consumption. However, whether we can use probiotics for medical application, so here, for example, to fight against the infections, it's not very much studied, even though there are some study. For example, here, there's a review paper, and they summarize the probiotics usage in wound healing. So here they have a lot of mode of action descriptions, how probiotic interact with the pathogenic bacteria. For example, how the probiotic can produce a lot of beneficial compounds, which allow probiotic to compete with pathogenic bacteria for nutrients, for space. And also probiotic can produce antimicrobial peptide, for example, to kill pathogenic bacteria. And also probiotic can produce also let's say growth factors and also other factors which can promote human cell adhesion, cell proliferation, or even modulate our immune system. So based on this background, so we started a research topic which is financed by our Swiss National Foundation. And the PhD student, Xuan, has analyzed different aspects of probiotics for this effect on, let's say, human cells. Here is human skin fibroblast. So here is the example. Don't worry, I explain you slowly. Huh? So look at this very left panel, and these are mammalian cells. You try to scratch and to produce artificial wound. Here's a scratch you can see. This is empty space. Indeed, this is artificially made wound. And then you incubate these cells in a medium, normal medium. Then you let the cells proliferate for 48 hours. And then the cells itself, they can proliferate, can migrate and proliferate, and then cover this wound, repair this wound. So here is the control, means the cells are exposed to medium. And then once we add probiotics in the medium, you can see this elongated lines, these are cells. You can see the wounded area can be much faster covered by the cells, means the cell migration, cell proliferation are accelerated by addition of probiotics. That's really nice. And then he also studied whether these probiotics can kill pathogenic bacteria. And here he used the Pseudomonas aeruginosa and Staphylococcus aureus. These are two typical pathogens in the wound infections. And then here, these are the agar plates, and then if pathogenic bacteria can grow, they form a film on these agar plates, this greenish colored film. These are pathogenic bacteria. Once in this hole, we load probiotics. Once the probiotic can kill the, let's say, pathogenic bacteria, and then you see a hollow, transparent zone, means this pathogenic bacteria, Pseudomonas aeruginosa, cannot grow in this area. This demonstrates probiotics have antimicrobial activity against this Pseudomonas aeruginosa. And similarly, they also show the inhibition zone against Staphylococcus aureus. And then we also use ex vivo human skin samples and made artificial wound and infected this wound with Pseudomonas aeruginosa. These are the scanning electron microscopy. You see the cells, bacteria cells on the skin wound without treatment. And then this is after treatment with probiotics. You can see the cells get somehow irregular, and also the membranes are destroyed, and some of the cells are even lysed. Quite nice. And then with this, I want to conclude this part, that we can say in the in vitro and the ex vivo study, and then probiotics exhibited very nice positive effect in antimicrobial activity and in promoting wound healing. But these probiotics were applied in a suspension. So, and the stability normally is not ideal for real application. And also the release of probiotics is also not controlled. It has, also the carrier material for wound management is also so far not very well studied. And therefore, in parallel, we had another PhD student working on this topic. And what he tried to do is he tried to encapsulate this probiotic, this green labeled cells probiotic in a hydrogel, and to reach several goals, like easy administration to wound and to provide a physical barrier to keep the moisture of this environment. And also this hydrogel should be biocompatible, not toxic, and most importantly, to reach controlled release of antimicrobial active and compound, all these beneficial factors for wound healing. So what did he do? He used two materials. One is gelatin. This is the biomaterial as part of our skin tissue materials, gelatin. And then he functionalized the gelatin with hydroxide, and then cross-linked with PEG, functionalized PEG. And then this functionalized gelatin and PEG can form a hydrogel through shape-based reactions. And this shift-based hydrogel allowed, this hydrogel to be injectable. Here is just demonstration. He loads the hydrogel with rhodamine B, which has a color, just to demonstrate this is injectable. And also this hydrogel is self-healing. You can see he cut this hydrogel in two pieces. And then when he put these two hydrogel together, they band together again. And then he also want to see the gelation time. So it's just very rough short video. He tried to mix these two components together. And then after puts into one tube, he tried to use vortex and to mix them well, to homogeneously mix them. And then within 60 seconds, this gelation already took place. And then he made a further the material characterization of the formed hydrogels. And here I just selectively present some features of this hydrogel. So this swelling ratio means this hydrogel put into aqueous environment, and then this can swell up to 160% within 24 hours. And he used 3 different pH. pH 6 is a normal our skin pH, and 7 is when we have acute wound, the pH is also around 7. When we have chronic wound, the pH is very alkaline, it's around 8 or 8.5. So this different pH just to mimic different conditions of the skin wound. And you can see this didn't differ, the swelling ratio didn't differ very much. And also he made a prolonged stability assay to see the weight change for 5 days. And then you can see the weight didn't change very much, and the different conditions, they are quite stable. And then he encapsulated the probiotic into this hydrogel. This green, these shining green dots, they are bacteria cells inside the hydrogel. And means he followed he monitored the the load of the probiotic in the hydrogel at the beginning, how much, then 24 hours and 48 hours. You can see the probiotics are viable in this hydrogel and increase the cell number with time. And then this is quantitative. This is just qualitatively impression. So how the probiotic survived in this hydrogel, and here is quantitative data to see the viability of the hydrogel. He loads the probiotic with different concentration. For example, in one of the hydrogel, he loaded around 10 to the power 7 viable cells per ml. Hydrogel, and this is 10 times more cells in the hydrogel. So you see with different time points, for example, the low amount of bacteria with time, the cell number increased from 0 to 24 hours, and then they maintained more or less stable. And also with a higher number, the cell number also maintained more or less stable, means the hydrogel is not toxic to bacteria cells. And then he also analyzed the release of the probiotics in this hydrogel. He put the hydrogel into PBS buffer. I don't know whether you can see this hydrogel here in this a little bit white color in this transparent PBS buffer. So this is probiotic embedded into a hydrogel, and this is the control, means probiotic in a liquid in this PBS buffer. So this is after 24 hours, the bacteria, the probiotic proliferate into in this medium, and then the medium in the PBS, and then the PBS medium gets very turbid. And then after 24 hours in this medium, you can see when the probiotic is embedded in the hydrogel, the medium is not very much, it didn't change the turbidity didn't change very much. And then he also made quantitative analysis. So within 24 hours, there are not so much probiotic release from the hydrogel. This is very likely due to the cross-linked polymer network, because we also measured the mesh size is around 14 nanometers. It's far too small for bacteria, because bacteria normally has a size around 2 to 3 micron. And then he studied the hydrogel, whether they are still active against Pseudomonas and staph, as I mentioned at the beginning. And here he did first to see the qualitative analysis. You see this inhibition zone I explained at the very beginning. So this has demonstrates the active antimicrobial activity of this hydrogel. And then he also analyzed, quantitatively analyzed the pathogen load after treatment with hydrogel. So you can see it's really this hydrogel and probiotics encapsulated into the hydrogel can really reduce pathogenic bacteria dramatically, almost to zero. And then he also analyzed the cytotoxicity of this hydrogel, probiotic load hydrogel. And here you can see this is blank hydrogel, the no cytotoxicity. And then when encapsulates probiotics in 10 to the power 6 or 10 to the power 7, 10 to the power 7 viable cells per ml, and 10 to the power 8 viable cells per ml. None of this showed any cytotoxicity. And then he also analyzed the hemolysis and hemocompatibility of these hydrogels. So these are negative control. This is tritone X. So this with probiotic encapsulated hydrogels, as no cytocompatibility is still preserved. So quite nice. This the progel showed no cytotoxicity and excellent hemocompatibility. And then finally, he also analyzed this bioactivity of this progel with ex vivo human skin samples. Here, just to show how this experiment were performed. This is a piece of human skin, and then we artificially created a wound which has a diameter of 6 mm. And then he loads pathogenic bacteria, means artificially infected this wound with Pseudomonas aeruginosa. And then afterward, at the same time, load and then put the progel on top of this infected wound. Then after 48 hours, you see what happened. So the control means no any gel, just a control with medium. You can see this is a staining where you see the bacteria cells on the surface. This is a wounded tissue. So you see this bacteria on the surface quite a lot. And with the gel without probiotic, you still see some quite some bacteria on the surface. Even though you compare to the control, you have a feeling you have reduced bacteria. This is mainly because of mechanical removal of the bacteria from the surface. And then once you have the progel, meaning hydrogel loaded with probiotics, and then all the pathogenic bacteria are gone. So here we clearly demonstrated the progel can inhibit the biofilm growth on these ex vivo skin samples. And then some summary and conclusion, and also take-home message. So this living material containing probiotics was successfully fabricated with desired mechanical property. And also these fabricated hydrogels showed very nice viability inside the hydrogel. And also this hydrogel allowed clear antibacterial activity in vivo and ex vivo. And this hydrogel also showed hemocompatibility and cytocompatibility with human fibroblast. And even though we have achieved quite nice work and the result in the lab, but we still need to understand why this positive effect from the probiotics and how they interact with the tissue cells. And later on, also for translation to a real application, there are still a lot of steps to go. And for this, I would like to conclude. Before I conclude, I still need to acknowledge our founding agent, like Swiss National Foundation, and also indeed I also mentioned some part which is funded by a EU project. I want to thank Dr. Zhihao Li from University Hospital Zurich for initiating this project together with me, and also Dr. Volker Schmid and Baharak Fleury from our local hospital for providing the human skin samples. And I thank you for your attention, and I'm happy to receive any questions.
Thank you very much, Chin. You are right on time. I believe we do have some questions on the chat, but I could start the questions so that people have time to type in their questions. Could you, Dr. Chin, could you explain what is going on? I mean, how does probiotics inhibit other bacteria? Because they are both bacteria. Why does one bacteria kill the other one? That would be nice to understand. And also, how do you see the role of hydrogels in encapsulating these bacteria or the probiotics? Do we need them to be released at one point? Because it seems like the gels are quite hard, and then the bacteria cannot move out. Or so these are the two questions, and hopefully others have more questions.
Yeah, thank you, Sabia. So first question about a mode of action, why probiotic can kill pathogenic bacteria. So this is indeed probiotic, like the one we choose is lactobacillus. They produce a lot of acid. So this lactic acid. So this acid can kill some pathogenic bacteria, and especially Pseudomonas aeruginosa is a typical skin pathogen, and they are very pH sensitive, especially to acidic conditions. And there are also other reasons in addition to this pH change, acid production. And for example, the probiotic can produce antimicrobial peptides. This, some like indeed commercially available antibiotics. Some of them are also antimicrobial peptides. These are produced by yeast or by bacteria. So probiotic can produce some antimicrobial peptide, which can also kill bacteria. So they also produce a biosurfactant, and then they can compete with pathogenic bacteria for surfaces. It's really very interesting. We are diving into, deeper, try to understand what are the components probiotic produced impacting the pathogenic bacteria. So this is first question. And the second thing about hydrogel, indeed, this is really good question, Sabia. And indeed, we are currently, because this gelatin-based hydrogel, they are bioresponsive. I don't know whether in this community I can mention this one. Recently, we just have a manuscript in revision. We want to report a bioresponsive hydrogel, means the gelatin, which can be degraded when pathogenic bacteria are present. And then the hydrogel is degraded, and then we can accelerate the probiotic release or even phage release. This is other component release. So this is just what I showed today. It's just one example. And also for translation, there are a lot of steps I mentioned. We can combine with the bioresponsive material or even modify the cross-linking degree to either accelerate the release or reduce the release. So these are, next steps we are trying to tackle.
I believe also the change of pH could trigger the release, and that could be one of the goals going forward, especially the wound when it changes pH. Thank you very much, Chin. Maybe you can go through the questions. We do have a few questions. We can start with, I think, Kanti Zasani. He has the first question. Do you want to read the questions and answer them?
Let me see.
Or I can actually read it for you, which is actually
Okay,
much
okay.
easier. So, so the question here is, what are commercially available probiotic wound care products in the market today?
That's also a question I ask myself sometimes, and I think the Petra can answer this question much better than me. I leave this question to Petra. Yeah,
Okay,
this is more commercial part. Yeah.
Okay, very good. So the other question here is, what are the bacteria being used? I think you partially answered this question before. What antibiotic is being produced by these bacteria, and how could this still affect antimicrobial resistance?
Yeah. This is also a very nice question indeed. So this what's the probiotic we use is lactobacillus mixture. We have 3 different lactobacillus species and strains mixed together, and we believe they don't produce classical antibiotics. As I explained, they produce acid, they produce antimicrobial peptide, they produce a biosurfactant, and they even can impact the colon sensing system of pathogenic bacteria. And because they have a whole, and they have they combine different mode of actions. That's why they can easily overcome the resistant problem. So that's not like a single antibiotic, which can provoke resistant consequence. With probiotic, you have different mode of action. So if, let's say, bacteria generate resistance to antimicrobial peptide, then they still have other factors like a pH, or they have other biosurfactant or colon sensing inhibitor. So all these combined together, then for the pathogenic bacteria, very difficult to make them to generate resistance.
Very good. I saw there was a hand raised. Okay, there's another question I can see. Will there be a potential to use textiles, for example, non-woven structure instead of hydrogels, or is there any study regarding that?
And the textile is also indeed we also intended, because the wound dressing is a 2 different material for sure, a category. Textile dressing, yes, maybe we can think about. I'm not aware of textile encapsulating probiotics for wound healing. Maybe Petra, you can say something on this, but I'm not aware of this one. But I could imagine if you have textile like liquid core inside, because you need to keep probiotic alive. Maybe inside the core you have aqueous or moisture or liquid environment where you can keep the probiotic alive. Then outside you have like a classical fiber, and then apply this one.
I believe there are these electrospun fibers which could then be loaded with probiotic, but that would actually mean that we need to spin under aqueous conditions, and temperatures have to be reasonably low, I would say. Yeah.
Yeah, you can make wet spinning. Nowadays wet spinning is also quite popular.
Yeah.
I
Yeah,
think.
but then we have to be careful. We cannot use solvents. I would say most
Yes.
polymers need
Yes.
solvents, and then that would be a problem. But I think this is some technical challenges which could be addressed at one point. There's another question: can a Band-Aid be impregnated with such products to improve their care?
It's I don't know this Band-Aid. What kind of materials are they made?
I think there is this membrane, and then below the membrane there are some, there is this active part, or I think the buffered part, and which are like, I think if I just imagine, there are different kinds. So you may think of a hydrogel which could be put at the. I think this white part normally which covers the wound itself, I would say.
Yes, I think that's indeed. I think there are some reports. This is adhesive membranes or something. I could imagine if they are if you can offer, aqueous environment to keep these probiotics alive, you can definitely use it for this purpose.
One last question and then move to the next speaker, I would say. Pedro is waiting. Do we have FDA approval as an antimicrobial agent for this probiotic? I hope that is what it means.
Yeah, no, I don't think it's any FDA approval, but FDA approval for oral, probiotic usage for oral application, you can administration. This is,
Okay.
you can buy it in the market. But for medical application, if you make a claim, for wound dressing, or I'm not sure. I don't think this already.
Do we need an FDA approval, or it's not needed? Probably Pedro.
I think you need.
You need. I think Pedro is going to answer this. He's nodding his head. So yeah. So we will look forward to his presentation. So Chin, I think we will now stop the questions. Maybe we will come back to you later if we have more questions on your topic. But I really thank you for your presentation. It was very clear. It was also quite informative for me. I think there is a lot of potential for collaboration here at Empa and hopefully with you guys there, I would say. Thank you very much. So let's move to the next speaker. Pedro, you may want to share your slide. Let me introduce you. Mr. Santos is actually a laboratory manager at HeiQ, and he is based in Portugal, where he is working on the development and validation of functional textile finishes, particularly in the area of antimicrobial and anti-allergenic, I would say, materials. He's a biotechnologist and he has been very much involved in implementing and validating testing methods for antimicrobial efficacy. I think he has been also working on different standards, which also includes our ATCC standard development. And yeah, so I look forward to the presentation.
Okay, thank you for the nice introduction. Thank you, Sabia and ATCC Group for this meeting and organizing the meeting. And thank you, Dr. Kuhnrand, for such interesting presentation. It was a reminder of my academic years, basically. So I'm here to present. Can everybody see my screen? I do so. So I'm here to present a little bit of HeiQ and how HeiQ is now embracing, let's call it nicely, the gift of Mother Nature on the development of new technology. So let's start with a brief introduction of HeiQ. Sorry. Seems not working. Okay. So HeiQ is a Swiss company founded in 2005. It was its 20th anniversary last Friday. It was founded by Murray Height and Carol Centonze after a hike on the Swiss Alps, in which after several days hiking, their clothes were starting to smell, and they decided they need to improve not only their life, but the life of all the other users. They started by developing some anti-odor products and move on for different functionalities. Right now, HeiQ, the core that makes it move forward is new innovative materials, and they do this to improve the life of everyone. And we have two major business units: the HeiQ Advanced Materials, in which I'm integrated, and HeiQ Life Sciences. We also have three venture units that I will not delve too much into these ones, but if you need any additional information, I'm willing to provide you the contacts and make connections to the right people. HeiQ is currently spread all over the globe. We have 14 offices in all the continents with 7 manufacturing sites. We have a tapestry of people in our corporation with over 30 nationalities. We have 200 innovations spread across 11 patent families. And since early on, we are partnership with several brands that most of people are aware of their names. We try to develop our products taking consideration that we only have one planet, and so we need to develop new sustainable and close to, I wouldn't say 100%, but close to bio-based materials. Our products are certified by different standards and regulatory systems. As I mentioned, we are a company very focused on innovation, and for that we have another innovation advisory board on all that are in different universities spread across the globe with different backgrounds, from nanomaterials to biology and virology to a materials engineer. We currently have a partnership with over 30 universities all over the globe. We also have mentorship programs with PhDs and bachelor's students. Right now in Porto, for instance, we have two master's degree students developing their master thesis with us. And we do this because universities are the main source of innovation of the great minds that we have in science, right? So we are spread across the globe, manufacturing sites in the United States and Switzerland. We then have innovation hubs in Portugal, the United States, Switzerland, Germany, and then we have a lot of distributors that are providing support to our mills that work with the brands. So I'm not going to delve too much in this part because we already made the introduction of what are the probiotics. Just a little reminder. So probiotics is a definition that was first developed in 2001 by a joint force between FAO and World Health Organization. And this was later reiterated in 2013 by the HeiQ group. Probiotics are basically any microorganism that on the host provide a health benefit. We know probiotics in everyday products from kombucha, for instance, kimchi, from food supplements. You also have probiotics on skin creams, in shampoo, in cleaning detergents everywhere. Prebiotics are, like our marketing people like to call it, are lunchbox that provides like I wouldn't say a doping agent, but they enhance the way the microorganism will work on the host. It will promote and stimulate the growth and the activity of these microorganisms, and in parallel, they are also beneficial for the host. They can reduce infection, they can improve our cardiometabolic health, and can indeed interact with our immune system. And symbiotics is this was stated by HeiQ as a conjugation between a pre and a probiotic. So when you have a probiotic and you provide him with required substrate, they increase and they multiply. So a symbiotic is the combination of the two. We are currently using probiotics and symbiotics on several fields in the industry, in pharmaceuticals, and in textiles, which I will later delve. They have a high sustainability, since you can even use renewable or waste products from other industries to promote their fermentation. They have an environmental friendly characteristic, since they are, as we are talking about microorganisms, they are readily biodegradable. Right now, they may not be the most cost effective, but on the long term, the prediction is with the increasing development of the technology, this will be a cost effective technology. And also, they are since they are weaving, and basically they are placed on the host, and due to their metabolic process, they are releasing substances that have targeted functionalities. And these targeted functionalities can be explored by different applications. Now, how is HeiQ using this new paradigm? How are we embracing the advantages of Mother Nature? A little background: in 2021, HeiQ acquired a Belgian company called Crisol that had a lot of background in using the probiotic platform to develop new cleaning agents. They developed functionalities for air filters, for automobile applications, for homes, etc., for water cleaning. And we use this technology to develop our probiotic platform to be applied on textiles. We currently have two major technologies: HeiQ Allergen Tech and HeiQ Skincare. Allergen Tech was our first development on the probiotic platform. And what was the main goal? And for me, this is very relevant because I suffer from this every spring. It's allergies. You know, we suffer from the presence of allergens, and on our household, we have a lot of presence of allergens, mainly from our pets, from cats and dogs, from the pollens that we bring from outside, and also one of the major ones is house dust mites. Their shells and the feces they release, they have allergenic reactions to some sensible people. And what happens to everyone that is exposed to an allergen is that they develop a sensitization to the presence of this protein. The allergic symptoms are well known: is cough, red eyes, you name it. Not very nice to have. And I suffer this a lot, is that when you have a runny nose, for instance, you suffer on your sleep quality, and this will impact on your performance throughout the day. In some cases, this can develop into some pathologies like rhinitis and asthma. In our households, and I'm using data from the US because this technology was first developed in aiming for the American market, is that 90% of our bedrooms is targeted by a lot of allergens, and 73% of the three more detectable allergens are at a high and elevated level that can produce the nasty effects that I previously stated. And the issue is that some of these allergens are named aeroallergens. This means that they can, by a simple movement, by a draft in the wind, an open window, by the simple movement while you are sleeping, can make these allergens go airborne. And when you breathe in, they enter your respiratory system. Take into consideration that we spend at least 6 hours a day on your sleeping. You will have an intense exposure to these allergens. Therefore, you will suffer a lot from the allergens effect. So the solution that we developed in HeiQ was HeiQ Allergen Tech. It's a symbiotic protection. This is a technology that is used to reduce the exposure to allergens. It is a technology that uses naturally driven sources, and it's these are symbiotics entrapped on a biopolymeric matrix. This is a picture of a SAM picture that we took of an application on a cotton fabric. How this works? So we have our symbiotics, so the probiotics plus the lunchbox, the prebiotics, entrapped on these biopolymetic matrix. And due to their natural processes of their metabolic process, they are releasing enzymes. These enzymes can then digest a large group of these allergens that cause the nasty effects. And how did we validate this? We had a partnership with a German laboratory that's called BMA. BMA is a laboratory with over 25 years of experience in this area of the allergen testing. And what they are doing is performing an ELISA testing. So it's a enzyme-linked immunoassay test. Briefly explaining, they inoculate our treated fabric with the target allergen and see if there is a reduction of the allergen after 2 weeks of incubation. Right now, we can achieve over 96% of reduction for house dust mite. That's the major allergen present in our bedrooms. And then we have satisfactory levels of reduction for both cat and dog allergens. Allergen for me is, I'm more of a technical guy, is not so much satisfactory, but we are working on it. We are also doing the validation in our laboratory. We simply measure the amount of probiotics that we have on our treated product. And for that, we use a simple European Pharmacopoeia protocol for determination of the germs. We extract, so we have a treated textile. We extract those probiotics using a homogenization methodology. And we incubate to assess the number of bacteria, of probiotics. What we found is that if we have at least 10,000 colony forming units per gram of textiles, we can achieve at least a 96% reduction on the house dust mite allergen. It's quicker and also cheaper method to validate the application of our technology. We did several durability testing after washing, and we maintain a good probiotic counts on both cellulosic and synthetic fibers. We also have some testing after 50 washes for cellulosic fibers, and this is great. This tells us that the treatment that we are applying to the textile is not only effective, but is effective in the long term, in the long run. If you think that this can be applied, for instance, in hotel products, in bed linens in hotels, which are subjected to a lot of washing, this is a major point for us. And also regarding the durability itself, in terms of time, not only wash, we did the first industrial trial. This has 2.5 years, but is almost close to 3 years. And we do some quantification during this time. And what we are seeing is that the numbers of the probiotics that we have remain stable after application. There are some slight decrease, but this is almost this is not relevant statistically speaking. Our technology was also certified by the Allergy UK seal of approval, which is a great for us. Where it can be used, I already mentioned in hotels product, but on household items as well. So everything you find in your bedroom. So mattress covers, pillowcases, pillow covers, rugs, etc. Basically, wherever you think there could be a possible presence of dust mites, and whenever your pets decide to jump in. Taking what we learned from the allergen tech, we developed the symbiotic, the skincare. That's a textile finish to promote the symbiotic balance and boost the skin microbiome. So, as you may be aware, skin is our largest organ, and it's our skin that protects us from the external aggression, from exposure to the sun, to the presence of microorganisms, to simple wounds, etc. And we are often judged by people by the way our skin looks. And when you have a shiny, smoothless skin, it is perceived as more attractive than one that it's not. And our skin is in our skin you have lots of trillions of microorganisms that live on a balanced microbiome. However, when you have an aggression, when you have fluctuations on your hormones, depending on the lifestyle of the person itself, if it smokes, if it has a poor diet, you can have an imbalance of your skin microbiome, and this will lead to dysfunctions of your skin. This can be some simple as having more sensitive skins, having it more dry, or the appearance of pimples and eczema and rosacea. Also, it is often skin is often washed with soaps, and this can be an aggression to the skin by removing our natural oils and causing an imbalance of the bacteria. Our HeiQ skincare, what it does is promoting a healthier and more beautiful appearance, just like any skin cream with probiotics. It is a cosmetic treatment that is delivered by a textile that is close to the skin that will promote the balance of your skin microbiome, resulting in a more attractive skin appearance. Like our Gentech, it is a product that is bio-based polymeric matrix that will protect our probiotics and promote their slow release while they are in contact with the skin. So this is it's a patent pending. We are still waiting for approval for a global application, and we also have, like in our Gentech, we have durability testing in which we can see that we can ensure optimal probiotic counts up to 40 home wanderings. This, of course, will need to have a cross-linker to ensure durability after wash. Our products are all non-irritant to the skin. They have been cytotoxicity tested. They have been tested for irritation, and they are using ingredients listed on the cosmetic list's international nomenclature. Besides, we also see that the application of our product does not impact other properties of the fabric, such as hand feel and the color. How is this working? I already spoiled. Basically, you need to have a close to the skin textile, and upon direct contact, you see there is a migration from the treated textile to the skin. We did this used by a wear trial with over close to 70 people, in which we used contact slides, and what you saw was that people that use skincare treated articles before application, they had 0 to none. After they had to clean with ethanol, they had 0 to none bacteria on those contact sites. After using skincare treated articles, they had an increase in the proliferation of the microorganisms. And we also, in partnership with the University of Porto, we identified by PCR and proved that before the application there were none probiotics, and after use of the treated article, there were probiotics from our product in the skin. Where can this product be used? It is used mostly on textiles that are close to the skin. So every base layer shirts that you have, compressions. So basically, all that is polyamide is very use this kind of product because you need to have a close to skin condition. For instance, having skincare applied on a blanket, probably it's not the most efficient way, but on a silky pajama, for instance, is the best option to go. Both our products, skincare and our Zentech, as I mentioned, they are bio-based technology, and they are one of the products that have a high sustainability profile and have a biodegradable certification. Both of them are Ocotex suited and meet most of the regulatory standards that we have worldwide. And the fact that they are naturally derived and use waste from feedstock, for instance, they confer a sustainability profile to our technology. Sorry, and that's it from my side. So thank you all.
Thank you, Pedro, for this nice perspective on industrial applications. I believe we do not have any new questions, but I do have some questions regarding your presentation. So you mentioned one of the application you guys have been looking at is against allergens. The question I would have there, what is the mechanism behind this deactivation of these allergens, I would say, with your treatments?
Okay, so what we have a probiotic mixer. I cannot, unfortunately, I cannot divulge the mixer itself, but what they are doing is they are naturally releasing cytokines, and these cytokines, they interact with the allergens.
Very good. I see there's a similar question from Chin. So what is the mode of action of probiotic in reducing the allergen? I think you just answered the question. That's the second question from her is, what is the washing condition for probiotic containing textiles?
Okay, we usually validate according to ISO 6330 4G, so 40 degrees gentle centrifugation. But
okay,
we already had some testing at 60 degrees, and we still have optimal durability.
okay.
I cannot recall. I don't think it's for 40 washes, but I think it's like 20 or so. I would have to check.
There was another question I had because you mentioned a lot of these applications could be bed sheets, upholsteries, I would say in hotels and stuff. But these textiles need to be ironed or at least needs to be crease removed, I would say, huh? So imagine, you have somebody going with steam or with iron going over these textiles with probiotics. Do they not get deactivated with temperatures are quite high, huh? When you do these treatments?
They can be affected. They are very much affected.
Yeah.
But as long as you have at least a log 4 after these processes, you are certain to have a good allergen reduction. We already had some, for instance, for dry cleaning.
Mm-hmm.
We tested for dry cleaning. There was a big decrease, but it was still in the log 4 something, 430, if I'm not mistaken.
Okay. There's this third question from Chin. It was, do probiotic containing cosmetics need authority approval? Huh?
This is a very complicated issue because This would fall under compliance discussions.
Okay.
But we are trying not to say it's a medical. We are not treating any kind of disease, of skin disease. What we are doing is preventing them from happening. Is different.
Very good. Can you kindly go over the testing recommended to evaluate fabrics treated with skincare?
Okay, right now there is not a standard method, nor a TCC, nor ISO method, but what we did was a wear trial. We basically, you have contact sites that are used, for instance, to measure the contamination on surfaces in food industry, and for instance, and what we did was we asked 70 volunteers to first wash with soap, water and soap, clean the surface area either on the shoulder or the outside parts of the thigh with ethanol, and put a contact site. This would give us the number of the presence of microorganisms that you have prior to the test. Then they would use the fabric, the treated fabric, either during a physical activity or during their work hours, whatever they were appointed to do. And afterwards, we would remove the fabric and place another contact site on contact with the skin. What you will see was that before using the garment on the treated articles, on the volunteers with the treated articles, you would see contact sites with little to no contamination. After using the treated articles, you would see an overgrowth on the contact sites. This said that there was a repopulation of your skin after cleaning. But now this leads the question, is this probiotics or is this a normal skin microbiome that is repopulating? And that's why we contacted the University of Porto to do PCR analysis. And through the identification of the 18S, they detected several strains that were on the skin after use. And we saw that there was over 80% of those bacteria that were present, over 80% of the bacteria that were identified were the ones present in our formulation. So yes, there is no international standard to do this. We had to develop the methodology ourselves.
Okay. I think there's a follow-up question which says, can you indicate the appropriate AATCC test method that would be used to measure the antimicrobial properties of treated textiles?
We don't want antimicrobial testing.
Yeah.
Because the issue here is that we never tested for antimicrobial efficiency.
Yes.
But like Professor Kunran said, you have a quorum sensum of your surface. Yeah. And what in theory what would happen is if you have a population of your good bacteria, you will not have your bad bacteria proliferating on that area. Mm-hmm. But this is very complicated because this would fall on a biocidal claim, and we do not want that, because we know our product is cytotoxically safe. We know mm-hmm. It's not skin irritant, but having this bacteriostatic evaluation could jeopardize the claim.
Okay. There's another question on the potential. I also had the same question. What do typically these treatments cost per kilogram or per square meter of fabric? Do you have any indication?
I don't know, because I'm the guy in the lab with the bacteria. I leave the quotation for the salespeople.
Yeah, okay.
If you want, I can get you in contact with our sales force.
Okay. But I think the person can directly contact you, I'll say, on that topic. There's a comment that AATCC RA31 technical committee could be interested in assisting in developing a standard if it is needed. It's probably also AATCC's mandate to come up with standards if there are new technologies available.
That will be great, because not only for skincare but also for allergen tech, we had a lot of problem because there was no allergen reduction protocol when we developed the standard. And I think we are in 2025. It was in late 2023, so 2 years after we started selling our allergen tech, there was an ISO standard developed that is ISO 4333. But the issue with this standard is that it is directed to textiles treated with heavy chemicals, heavily chemically treated textiles like metals and so on, that have a quick action against the allergens. And our technology, it's not like that. It's a probiotic based, so they need time and they need conditions to proliferate and release the enzymes.
Okay.
And
I mean,
that being short, if in any case, if RA31 would like to develop a testing method, we would be very pleased.
Okay. I mean, you presented two of your own technologies, but are there competitors who are also coming up with similar or different ideas in the market?
I think so. There are some competitors in the market, but there are different, they have different functionalities and mode of actions.
So it says, the comment was, please join the AATCC RA31 meetings in May. So if you want to be part of this standard, you probably would like to contact AATCC on this matter. It says that there is another question. You use cross-linking to cross-link the probiotics to textile to enhance durability. On the other hand, you mentioned the bacteria released to the skin to fight the pathogenic bacteria. How is this? How is it released from a cross-linked surface like that? Yeah.
Okay, because what we are linking to the textile is not the probiotic itself. It's the biopolymeric matrix in which they are entrapped. Imagine, like, I would say, a hanger. You know, you have your coat on your hanger and you place your hanger on your wardrobe, and you can either ways remove things from the pockets of your jacket that is in the hanger. I don't know if this. I'm very bad with metaphors.
No, I agree. If the gentleman has more questions, they can always come back to you. I just want to go through all the questions. Is HeiQ product used for filters in ventilation application? Huh?
Yes, it is. I'm not in that project, but I know there are some brands of ventilation filters using our technology.
Very good. I think we have gone through most of the questions. They even invite you to join this meeting, which is free. I would say to join these committee meetings. I believe we have gone through all the questions. If anyone has any last remaining questions, feel free. If not, I think we have a. Okay, there's one more question. Why fabric skincare if there is skin cream? It's a valid question.
It's a valid question. I think here the answer is why not? Because we want to be innovators, basically.
But,
I think here it is more. I'm not a skin cream user guy, but I think the selling point here is why spend 15 minutes
creaming yourself when you can just put on a textile, and which you do on daily basis, I would say. So yeah, there's always a good argument there. So I think I would like to stick to the time here. I promised 1 hour 20 minutes, and we are almost at the end of 1 hour 20 minutes. So it seems like there are very interesting technologies which are in R&D as well as in commercial development, and I see where we are moving towards. I would say where we are moving to in future. So both our speakers did an excellent job in presenting their perspective, academic as well as industrial. and I learned a lot. I believe you all too. Let's thank our speaker again. It was a very nice discussion. I hope this topic will be discussed more and more in future. I think we do have some few slides which Shania is going to present. And this meeting is recorded and it will be available for the AATCC members to have a look at it. And if you have any questions you would like to ask the speaker directly, so feel free to do so. We will be happy to connect you guys. But I was made aware that the slides will be not shared, but the recording would be shared among the AATCC in the AATCC community. So thank you again for all the involvement of all the audience and also the great speakers. Shania, would you like to go ahead and do the final few slides?
Yes. All right. So, yep, thank you again to the speakers for taking the time for this special presentation. Just wanted to go over that our volume 100 manual is now available. So you can order this on our website. So we have some methods that are updated, and it's our special 100th volume. So you have the nice red cover as well. So that is available for you to order. Here are some of our upcoming events that you should mark in your calendar, and you can go on our website as well. Some of these registrations are already open, and wanted to just highlight that there. And then if you are not already subscribed to our newsletter, that's a great way to get connected, to see all of the industry updates as well as upcoming member events. Then if you have any additional questions from AATCC, you can contact the membership team. You do membership at aatcc.org. That gets you to all of us, or here's all of our individual contact information as well. So that wraps up just all of the brief outro AATCC items, and wanted to just thank you everyone for joining us today. That concludes the presentation. Thank you, everyone.
Thank you, Shania, for helping out. And the AATCC team. Thank you very much, guys.